So for obvious reasons, I'm trying to be punctual in this part. My eye is going to be on that clock, don't you worry. So I told you what we're going to do is we're going to extend from this understanding of, initially that came from animal collectives, that then we found actually a very simple model, a very generic model of social influence, whether it could be neural influence or other types of influence, such as humans influence each other, potentially could have commonalities. Potentially there's this deep mathematical similarity across scales of biological organization. And I won't have time to talk about this paper today, but it was quite an important one from my perspective, because we started studying physical systems, specifically spin systems. Seems a bit over loud, doesn't it? And a bit interfery. Pardon me? Oh, you turned the volume up. Okay. If there's a problem, just let me know. And so this just again reinforces the view that we looked at a very different system, and specifically one that had been very heavily studied in physics, where spins can just be up or down, and they're interacting with the spins around them. And again, we found that adding the unbiased individuals improved the capacity for the system to magnetize, i.e. for all the spins to point up, or for all the spins to point down, which is effectively a consensus decision. And similarly, the proportion of uninformed spins that didn't care if they were up or down, that maximized magnetization or maximized the decision-making capacity, was also associated with a drop to time taken to make the decisions. So the uninformed individuals facilitated decision-making and also reduced the time taken to make decisions. So yet again, we would never have thought to do this. Having some spins in a field that they prefer to be up, some spins in a field they prefer to be down, and some spins don't care. And there's all sorts of interesting things about the coarsening dynamics. We can also prove this. Andrew and Emmanuel, can prove this result using coalescing random walks and all sorts of other stuff. But that's just to make the point that you can draw these connections mathematically. And why it's important for today's lecture is that Bill Bialek, a physicist at Princeton, had this beautiful paper in Nature that showed a formal connection between spin system models and the cortex, neural models and real data in the cortex. And the important thing that they made in that point they made in that paper is that this is not an analogy. Like, they were arguing that there's genuinely a connection. And so I started getting interested in how the brain makes decisions and wondering whether the collective dynamics we see in animal groups may give us some sort of insights into the brain. And that's what I want to focus on today. So when I wrote this in 2007, I was very cautious. There are so many differences. Surely it can't be that there's such deep similarities. It's too good to be true. There are so many differences in complexity of the components. There's differences in the selection mechanisms. There's differences in the physical nature and the spatial embedding of the systems. So here I was very, very cautious to draw a connection. But what I'm going to argue today is that I think we can and should go beyond the metaphor. I think there are deep commonalities in spatiotemporal computation, mathematical similarities, that unite different scales of organization. And if we're right, then this could be something really profound. And I'll show you the evidence towards that. We've only just started working on it in the last couple of years. So I'm going to argue that there could be a genuine connection between neural computation and computation at other scales in collectives. So how do neural collectives make decisions? Well, of course, the brain is vastly complicated, and we could think of lots of different types of decisions. So we're going to restrict ourselves today to focus on very common decisions, and actually very primitive decisions that must be made by almost all animals. And that's how they make decisions as they move through space. How do they decide where to go? And over the last 10 to 15 years, there have been a series of studies particularly on model organisms, Drosophila, zebrafish, and the rhesus macaque, among others, mice, rats, and so on. And they've all come to a similar conclusion in that the brain must come to a consensus regarding where to go, and that the brain explicitly represents information about targets in space or preferred directions in space explicitly in a vectorial way. That is the neural hotspot here. So this is called an ellipsoid body in the Drosophila brain. And isn't it beautiful? This is a donut-like shape, a torus. And on this torus, there's a bump of neural activity. And this neural activity here directly encodes the direction and the strength of preference of the animal to move in that direction. I find it really remarkable that natural selection has literally created a torus to solve this type of problem. And so you have this what's called a ring attractor network that's guiding the decisions. Now, if you look at the mammalian brain, they do not, to our knowledge, have a physical ring. However, it behaves like a ring attractor. Functionally, it has been shown that it is a ring attractor. It doesn't, so you don't actually need the neural groups to specifically to be aligned in a ring, but the connectivity in the brain can make it in the functional space a ring, even though it's not physically a ring. And similarly, the head direction cells in this case come to a consensus about where it thinks the animal is looking as it's moving through space. So really remarkable studies in the last 5 years. There's also equally beautiful, if not more beautiful, studies on bats by Nakamuranovsky that also shows that not only do they encode preferred directions and so on, but they specifically have, actually there's 2 papers back to back, one from Nakamuranovsky in bats and one from a Japanese group on rats that showed that these animals specifically encode vectorial information about other individuals. Not just about objects, and they can differentiate between, in Nakam's paper, they also had an American football going through space, and the bats didn't care about that. But if it was a bat going through space, they paid attention and had a vectorial representation. Even the Drosophila, if you put an object between it and its preferred direction or a target for a brief period of time, a few minutes, it will still have a memory of where the object is. So I got really excited when these discoveries were made because I've been thinking about vectorial integration, but thinking about multiple individuals within a group. But the brain, of course, is a collective, and in some ways is easier to work with because you can ask where does the individual want to go rather than where does the fish school want to go, which needs a lot of space and training and so on. But to do this is really hard. And why it's hard is, as you can imagine from the previous lecture, in my mind, what I was excited about was the possibility that the brain may exhibit similar bifurcation behavior that we'd previously seen in animal groups, right? And as soon as you see a bifurcation, remember if I added one extra individual in the simulation and instead of it being 50-50, they almost always went in that preferred direction? Well, if I'm doing experiments in my lab, how can I know I'm completely controlling for all of the environmental factors that may break a bifurcation, that may break the symmetry? There could be an airflow that I don't know about, there could be light, there could be magnetism. But in virtual reality, we can very easily control for these factors without having to rearrange the entire lab every time. And there's other benefits too that we can get to. So this sounds high-tech, and it is, but it's actually based on a very old illusion. It's called an anamorphic illusion. And I can tell you right now that this tape does not have a volume above the table. And I can tell you it's an illusion, but your brain still believes it to have a volume until the illusion is broken. And if the tape goes even roughly back into the correct position, your brain will pop it back into volumetric coordinates. Similarly, this shoe does not have a volume, right? So even if I tell you, your brain believes it. So if a human can be fooled in this way, surely an animal in a group can. So put another way, if you track the camera and reconstruct the visual field from the perspective of the camera, which is like an animal, then it can move in behind and fully immerse itself with an arbitrarily complex three-dimensional environment. Okay, does that make sense, the concept here? And so we've created a whole suite of these virtual reality environments of increasing sophistication. I'll predominantly talk about the one that we used for fish today. So we have 4 cameras that track the animal in 3-dimensional space. The animal is in a bowl. It's about 35 body lengths across. We control, of course, for the distortion of light on the surface of this hemispherical bowl. So the projector bounces off a mirror, and then the world is always displayed from the perspective of the animal. So it looks weird to us, but it always looks like that shoe did, as if it's volumetric from the perspective of the animal in the arena. And so here's just a simple pillar. And of course, there's nothing actually there, but the fish believes it to be there. It believes as if there's an object there. We also can create virtual objects, so holographic conspecifics that can appear to be in the volume, swimming with the real fish. And I should state that what we're using here, this is why they've got such big eyes, these are juvenile zebrafish. We use them for three main reasons. One is that they're a model organism for neurobiology. And when they're juveniles, they're transparent or semi-translucent, more or less. And so they're amenable to neural imaging. That's one huge benefit. Benefit number two is that because they're so tiny, less than a centimeter long, it's like they're going through syrup. So they're covered by a thick, sticky boundary layer, viscous boundary layer of fluid. So the lateral line is completely dominated by self-generated motion. They can't feel other individuals around them. They tend to swim also centimeters apart. There's no way. And also, there's no way olfactory cues, which is going to be dominated by diffusion, would have time to at least be used for schooling behavior. And last but not least, because they're so tiny, and because we need to suspend the weighted water above an open bottom for a projector, and we don't want this thing to collapse, the smaller the volume of water, the easier it is for us. So many different reasons why we focus on juvenile zebrafish.
And the juvenile zebrafish already exhibits the... Okay.
So I could repeat it. I think you could ask, does it already exhibit social behavior? But we'll see that in a moment. Sorry to rush you along, but my eye is on that clock. But yeah, so here you can see, for example, exactly what you're saying, a proof of principle. So here we have the virtual zebrafish just moving in a circle, and you can see the real fish schooling with it. It believes it to be there in the tank. 80–90% of the time, they will school exactly the same probability of them schooling with a real fish. So even though we're projecting just onto the surface, it believes there's this holographic fish in the tank with it. But of course, there's one problem here, which is, remember that the anamorphic illusion? I can't put two fish in one of these tanks. Because remember when the paper was moved slightly, suddenly the illusion's broken. So the illusion can only work for one individual, because we're presenting it from the retinas of one individual. But what we can do, and what we have done, is we build multiple systems. We're building 15 of these at the moment, and we can network them together. So this one can interact in real time with a hologram of this one, and vice versa. So we call this the matrix, because they're not interacting. So here you can see two individuals that are not interacting in the same physical world, but they're interacting in the matrix, in the same holographic world. And here you can see that we've taken great care, and I'm not going to go through all the details of this, but Liang, a postdoc in the group, has taken great care to show that the way they respond to others in the matrix, in terms of how they respond in terms of the speed and the turning, and this is just illustrative. Everything you could possibly measure is exactly the same as when the two fish are in the real world, which is not how they respond just to any object. So they really believe this to be another fish. In fact, the data in the matrix is cleaner than the real world because the fish are so tiny and transparent that when they get very close, sometimes the tracking system confuses them. And in the matrix, of course, we never confuse them because there's only one in every environment. So the data are even cleaner from the matrix. So here you can see four individuals. They're not interacting in the same physical world, but in the same holographic world. And what we're doing now, we won't have time to talk about today, but of course, we can give different information to each of these individuals very easily because we completely control that. We completely control whether there's a time lag, whether I can see you but you can't see me. All of these things are now completely under our experimental control for the first time. We've also developed virtual reality assistants also for flies, for Drosophila, again because they're a model organism. So it's on a pin that's connected by magnetism, and so it can freely move in a virtual environment. We've also developed a huge, it's the biggest we could fit through the door, motion compensating sphere. So when this desert locust moves, the sphere moves to compensate for its motion, so it can move in a never-ending environment. And of course, this is just for calibration, this checkerboard, but it's the only video that I had. So here the animal can even jump, and it will just be put back into place. So this virtual reality works really, really well for all of these organisms.
Can you repeat how you did with the fly?
The fly is just, you can just about see it on the left there. There's a pin glued to a very thin metal glued to the back of the fly. I mean, these flies are tiny. So this is an incredibly thin bit of metal. Now, to allow the fly to turn, it's really hard because of friction. And so we just have a magnet at the top that holds the pin in place, but gives just enough magnetism to hold the pin in place while being frictionless, so the fly can turn. So we're now building a system, so this only allows the fly to turn like this, right? So it can't do this or this or this or this, and it works, right? I don't know why we didn't do walking flies, that would have been simpler. So we're building, one of my PhD students, August, is building a virtual reality system for walking flies that we think is going to be more powerful. But another postdoc in the group is actually building a very cheap 6-axis force meter. So we can actually have flying animals moving in any of their potential directions. That's never been done before. And he's come up with such a simple way, clever, simple way of doing it. Previously, people have spent hundreds of thousands on equipment to do it. His probably cost about 20 euros, and it works. So that's, I shouldn't talk about it without his permission. So all of these systems allow us lots of benefits. You know, we have the complete control over the visual information. And so for zebrafish at this age, it's exclusively visual. For flies, the same guy that did the 6-force, he's also doing multimodal, olfaction plus vision. But here we just have vision. We can quantify inter-individual differences. Again, it's important, but not time to talk about today. But what is really important today is it gives us a unique capacity to determine how the brain makes decisions for 2 reasons. One is that we control the inputs, right? And so we can precisely control the experiment, which means we can explore parameter space systematically like you normally do with a model. But we can do that, again, with the real brain. And also, we can finally control causality. You know, if I am influencing you and you're influencing someone else and we're interacting, am I being influenced indirectly or directly and all this kind of stuff? That's always a mess, which is why I could only ever look at the first individual responding to the alarm. Now, of course, we can control who can see whom. We construct the network. We can control lots of things. So we think this is really important. And I think I probably forgot to put the figures in, so I'll mention it now. We're working with Armin Bal, a neuroscientist, who just got a big ERC grant yesterday to do this with me. We're also building a virtual reality environment inside a 2-photon microscope, so we can get full brain imaging, cellular resolution, the entire brain, while the animal is freely moving in virtual reality. So we can really test a lot of these ideas. I probably don't have those slides, and I took them out, I think, but I can show it tomorrow if we need. Okay, so this is going to be our tool. And just to remind you of the question is, how does an animal make a consensus regarding where to go when it's moving through space? Vivek was a PhD student with me, and he did all of the experimental work with flies, which he hated. He's now working on black buck in India using drone imaging. He also did all the simulations, which he loved. Liang did all of the zebrafish experiments. Dan Gorbanos is our black hole physicist. He did the analytic models, which I won't talk about because I don't fully understand them. And Bianca, I was telling people at lunch today that I cannot solve an equation, and it's absolutely true. Bianca is an undergraduate in my lab who still works with me, and she did all the locust experiments. Okay, so the idea is we're going to create a very simple model of this ring attractor. And we are not the first people to do this, by no means. This is a classic model in neuroscience. But amazingly, it has not been studied in the way that we studied it. So all it assumes is that there's reinforcement among neural ensembles that have similar directional preferences. So for example, if there's an object over here, this stimulates this neural group to fire, and that will also excite these ones and potentially inhibit those that disagree. So it's very much like an opinion dynamics model, where you tend to be reinforced by opinions that are similar and kind of repelled by opinions that are different. So this type up was also a bit like a reaction diffusion model, for those who know those, where you've got local excitation and longer range inhibition. The mathematics of all of these models is the same. And so it's really an aesthetic or a choice of the researchers how they model it. We happened to model ours as a spin system, because many of the researchers come from physics backgrounds, but you could easily model it in different ways and the results stay the same. And in the paper, there's a proof that shows that you can go back and forth between the spin system model and the neural field model. It's the same model. And that was thanks to Dan being able to show that, to prove to the referees that it's not crazy to use a spin system model to understand neuronal dynamics. But overall, whether you model it in our way or another ways, whatever you prefer, but it's always a neural ring attractor. And actually, after we published our paper, we knew the ellipsoid body exists in the fly, but we did not know really about the locust. It's probably ellipsoid-ish body, but the fish we had no idea about at all. So we predicted that it was going to have a ring attractor, as you'll see in a moment. But since this has still not been published, but it's on the bioarchive, they've actually found the ring attractor within the vertebrate brain. So it's not impossible that we also have a physical ring attractor. It could be something that's so deeply convergent in vertebrates. It's taken this long to find it in a zebrafish. So I wouldn't rule it out that it also is in higher mammals as well. So the general idea is animals consistently have to make decisions about where to go. And so what we're going to ask is there a common algorithm, a deep mathematical algorithm that explains it? So we just made this to accompany the paper. So our model makes specific predictions, and this will be reminiscent to you of the lecture this morning. So if the angle is small between the options, we predict that they'll adopt the average direction, but then the neural ring attractor exhibits a bifurcation. And so there's this emergent transition from averaging to decision making, directly inspired by what I'd seen in the flocking model. And so if you look at the analytic version of our model, and so this is basically showing this is the angle between the options. So this is small angle to large angle. This is neural noise. So you can see it doesn't really matter as long as the brain is not so noisy that it can't make any decisions. We can always prove that we get this result, whereby the brain should average until the animal reaches this solid line here where it has to switch. We can prove it has to switch to decision making and randomly choose between these two options. What we see in this gray region here is a hysteresis regime, whereby if the brain switches from averaging to decision making, it does not switch back, which is a good thing for a brain to do. In practice, we estimate the noise in the three systems we've studied to be 0.2, so there's quite a big hysteresis regime. So it's not going to be a very clean angle at which they bifurcate, because the brain, even the same animal, could bifurcate anywhere in this range of angles. But in each trajectory, we should see these bifurcations. So this is very much a stylistic picture. It's going to be more noisy in reality. So the animal should move towards this critical point, represented by the solid black line. The neural dynamics should fundamentally change, and the animal should then make a decision. Now, we wanted to test this theory, but it turned out that we could not find a single paper, not one in the whole world, where people had recorded how an animal moves when it makes a decision, despite the fact there are tens of thousands of papers of decision making. Almost always people put the animal in between the two options. Why not? Why would you think any differently? I'll come back to that in a moment. And because I'm not a neuroscientist, I can't go into the brain to look at the critical dynamics I expect at the phase transition, but we expect that this is an embodied control. What's happening is the animal moves, it changes the spatial representation, which changes the neural dynamics, which changes the consensus, in the brain which changes the movement. It's almost as if people before thought the animal just looks at the options, makes a decision, and movement is just the outcome. Movement is just how it gets there. We're arguing for something fundamentally different here. That is, if we're right, of course. So we tested this with fruit flies and with locusts. And so there are hundreds of trajectories here, and it's hard to see the overlap. So this is basically the overlap of these trajectories. So the insects, they start here. This is in meters. I mean, it's in virtual space, but it's representing physical space. And they're just moving towards two pillars. These animals, locusts and flies, are both very drawn to vertical objects because that's strongly associated with food. If you're in a desert environment, a vertical object could either be a perch, to shelter, or food. So it's very attractive. So clear evidence, indeed, that the brain does exhibit a bifurcation, or that trajectories are supportive of the hypothesis that the brain exhibits a bifurcation, which we were really excited about. Now, there's something else interesting here. So yes, this is important to understand why animals move the way they do. But there's also some very cool stuff that happens near a critical point. And you may have covered this before. So have any of you heard of susceptibility within these models? Yes, some of you maybe? So it's let me switch to the blackboard and I'll quickly explain where it was discovered. It was discovered again in magnetism, in studies of magnetism. So remember I showed you with the locusts, the magnet that was highly magnetic, and then you heated it up to the Curie point, and it lost its magnetism. So think of that again, right? So what happened is the magnet was strongly magnetic, but as you heat up the magnet, so this is the temperature of the system, there comes a critical point at which the magnet has lost its magnetism, right? So the order parameter here is 1, and here it's 0. Right? And if you actually plot this order parameter as a function of temperature, so this is the order parameter, it tends to be a very nonlinear transition between order and disorder. So this is what we mean by a phase transition here. And this is the critical point, 770 degrees in the case of a magnet. Now, imagine you want to influence the magnetic particles. Imagine this could be like opinion dynamics, or this could be particles in a magnet or fish in a group. If you want to influence which way they're pointing, at low temperatures, say I wanted to flip this magnet, instead of pointing this way, to point this way or to point this way. And I can use an external field to try to force them. But it's going to take a hell of a strong field to flip them. They're so locked in place. Now, say I try to do the same here with my magnet that's all over the shop. There's so much noise in the system. I again need a very strong magnetic field to get, it's like herding cats. To get them in order, to overcome the noise, I need a strong magnetic field. So at both extremes, I need to apply a very strong field to influence the system. But what was discovered is that close to, near the critical point, you need a tiny magnetic field to flip the system. And so this is measured, it's a measurement called susceptibility. How susceptible is the system to external influence? And susceptibility tends to actually increase to infinity for an infinite system, but for a finite system, it would have a limit. And it occurs near this critical point. So the system can be ordered, but also super sensitive. Or it could be ordered and incredibly insensitive, right? So if you imagine now, in my system here, I'm not talking about an external magnetic field, but I'm talking about, say, two different options. Imagine one option is slightly better than the other. It makes like the neurons fire a tiny bit more. But normally that's just swamped out by noise. What we predict is at the critical point, this difference will suddenly be magnetized. It leads to an ultra sensitivity that comes for free, just like it does in a magnet. It comes for free if the system's at a critical point. Now, previously, people have argued that the brain may be close to a critical state. But they've always been talking about tuning the system to a particular regime where it can exploit these dynamics. That's not what I'm talking about here. And still talking about undergoing a phase transition, but here it's a geometric change, right? There's no tuning. The brain can be any one of those noisy brains, doesn't have to be specially tuned in any way whatsoever. All of those brains should undergo this phase transition as they go through space, if we're correct, right? And so what we predict is at this point, and only at this point, the brain, even a small, noisy, silly brain, will spontaneously be like a supercomputer, or spontaneously be superly accurate. It's almost miraculously accurate, and then it becomes dumb again, right? And this will happen irrespective of the angle at which the animal reaches the targets. So we can predict this curve here. Now, in practice, we always do the experiments just in the middle because it's simpler. It's really hard to do these experiments, even in VR, because anything could break symmetry. But in theory, this should work from any angle. Okay, are there any questions about that? It's an important concept. That's why I think this could be really cool, this discovery. Okay, so we know that the perceived difference between options is important. If, you know, if you go to the pub here and you get offered one beer versus a third of a beer, and you've got, you know, which one you're going to choose? You're going to choose the one beer. But if they're both almost as full as each other and you're cheap and you really want to get the most, you might have to think about it a little bit, and then you take the one that you think's got the higher volume, right? The closer the options are, the harder it is to distinguish, and therefore, the longer you take to make decisions that are hard. So you can integrate more over time. This is true. These are facts of decision-making, the general facts of decision-making. But controlling for these, we think we've discovered a third principle, which is geometry. Is really important. And you would never think that before, right? If you think about, you know, what I said about biologists running experiments, say the black option is slightly preferred to the dark gray option. Why not start the animal in between them? Like, this is why everybody does it. Why not? But if you think about this theory, it would make a very big difference because the animal here cannot go through a phase transition. It's already well beyond the bifurcation point. It would have to leave and go back, which is an interesting prediction. Maybe they do. Or you can start the animal over there, the same distance, the same options, and everyone would have predicted the same decision outcome. We predict a very different decision outcome. And we're not talking a small amount. We're talking around 35–40% difference in accuracy. And so Lisa, the undergraduate in the lab, ran these experiments. And indeed, a locust does prefer the dark option. This conforms to previous experiments. But it's not very good at distinguishing unless it can go through the bifurcation. And then it's easy. Suddenly it becomes easy for the brain to do it. So again, it's supporting. So what we're now doing is we're using the 2-photon microscope to actually go into the brain to look for the critical dynamics, because that's where we can really, really prove it. Yeah, we have some questions.
So for different animals, does this critical angle somehow correlate with the field of view of this animal? Is it like, what is the logic behind this critical angle? Is it the point at which things start going out of your...
So because of the football, I actually took this out. But we could come back to it tomorrow, because the only way this can work is if the brain warps space into a non-Euclidean coordinate system, into what's called a spherical geometry space. That's the only way this can work. And it does it the same in the 3 systems that we studied. So yes, there's a bit more than I made on. Like the brain is actually warping space. So space is expanded in front, and but these animals are more or less 360 field of view. But that doesn't mean that they're using space in the same way. Space is not being used in the same way. I'm very happy to go back to that tomorrow when we have a bit more time.
In that experiment, the 2 targets are distinctively attractive for the birds.
This one here, you can see that the black one is a tiny bit more attractive than the gray one. And we know this, that they prefer a higher contrast, which is why we did this decision. Similarly, we could have used like the fish that like the yellow more than blue. We could have used like a yellow they like a little bit more than another yellow, something like that. It's got to, all it needs to sort of, because if this was an easy decision, the animals would make the decision like 100% of the time, for example, in both conditions. It's not going to be exciting. These things, this amplification needs to amplify something, right? So we need to start with something that's hard that can be amplified. which is why we went for sort of options that were actually, we deliberately tested different options until we chose one that was consistently slightly preferred in this case, and then we could move the animal to a different geometry and show that the animal suddenly makes much better decisions. But the distance between the options is the same, the quality of the options is the same.
And is there data regarding if you translate along the axis between the targets?
Well, it moves along and then you get the branching behavior. That's that.
I mean, in the case where you simply put there's a case where you put the animal exactly halfway between the targets.
Yeah, we always do. It's always to the left and the right is always equidistant.
If you move the starting point a bit closer to one of the targets, how does that affect?
We predict it doesn't. We predict it doesn't. We haven't. We're doing those experiments right now. So it was kind of. Oops, sorry, I've gone too far. Yeah, I didn't really cover that, but with the. So yeah, so this is us obviously starting them in the middle. But if we run the model, and maybe I'll show that in a bit. We get the same predictions irrespective of where they're coming from. So you can, for example, start down here and have the bifurcation occur closer to one of the targets, and it will still be very accurate. The neural tuning, this warping of space, is critical for this result as well, because maybe I'll come back to it with 3 targets, because this warping of space is really important for this and also for the 3 target choice. It's just easier to explain for 3 targets. So if we... Okay, so we think that this could be a general principle and could be functionally important. Okay, so almost all neurobiology and biology experiments occur to 2 targets. There's very few papers, a handful literally of papers that look at more than 2. Why is that? It's because traditional models do not scale well. Adding a 3rd option becomes really difficult. This model scales to any number of targets. So what do we predict if we have 3 targets? We now predict that the brain exhibits a double bifurcation. So the brain should break down the complexity of the world into a series of binary decisions. It requires an order of magnitude more data to look at this. That's why we've only done 3 targets so far. We'll move to 4 in future. But let's test this prediction. So if we're correct, the brain spontaneously breaks down the complexity of the world into a series of repeated binary decisions. And close to each bifurcation, the brain excludes an option and becomes super sensitive to the remaining options. Right? Again, for free is the prediction. But first we need to ask, well, do we have evidence that the brain indeed does this? So we did our experiments with fruit flies and with locusts, and indeed found evidence that the brain is exhibiting this behavior. It's breaking the world up into a series of binary decisions. We also, I know there's some mathematically oriented people here. I have massively simplified this story just because the general principles are so interesting. It turns out that the choice of the central option is actually a different type of bifurcation than the choice to the other options. One of the other things that relates to this non-Euclidean geometry, without the non-Euclidean geometry, so you can basically tune the neural tuning parameter, how you warp space, such that they almost always choose the middle option because the left and the right cancel each other. Or you can tune it the other way, that they almost always choose the 2 options on the outside and hardly ever the middle. Or you can tune it such that it's identical. And it turns out that tuning it such that it's identical is a smart thing to do, right? Because if there's a difference between these options, you don't want to be distorted by geometry. You want to be distorted by choice. And so we can also show that the region, the tuning, this non-Euclidean tuning, is exactly such that it's 50-50 between 2 options, 33% between 3 options. When you get to 4 options, only that particular neural tuning can solve the problem at all. Otherwise, you get locked in loops. So we think it's critically important that they're warping space in a specific way. Yeah.
When you say warping space, do you mean some kind of effect of the let's say, the lens of the visual system as well, or the internal representation of space that this...
The neural representation.
Oh, okay, so they represent space internally in a way that is non-Euclidean.
Pardon me?
So they are representing space in a non-Euclidean
On the ring attractor. The ring attractor is representing space in a non-Euclidean way. Now, of course, you could be right that the brain or the optics are warping space, and then the ring attractor would have to be tuned differently. But in the systems that we study, there's no, for example, fovea, or there's no high-resolution region. They have an equal resolution around. So for these systems, it's almost always going to be done on the ring attractor itself. But there's no reason you couldn't add more warping in, say, optics.
Because, I mean, we experience space through the as an Euclidean.
Yeah, no, we experience space in a non-Euclidean way.
In this sense specifically?
Yes. If you're making a judgment, if I ask you how far away my fingers are, and you make a judgment, it actually depends where in your geometrical space you make that. You're also warping it in front of you. And it's not just the foveation, because your foveation can be drawn to different parts. And it took us a long time to find that anyone else had even thought about that. They don't know it relates to this, but there's only 2 papers, and they're both with humans. Old papers that just show humans consistently misreport whether they're misrepresenting. They misreport distances in a consistent way to this. But I don't know. But yeah, I just wanted to mention that for those interested, we will have we're submitting a paper soon that will have a lot more of the details of this. Oops. But I did want to point out that it really works for any number of options, right? We don't have to retune it. It's exactly the same parameters for the fly and for the locust. It also produces all sorts of nice patterns for different geometries. And I'm all about patterns, so that was fun. Okay, but if this is a general principle, yes, these are 2 insects that are not very closely related to each other, evolutionarily, but they're still insects. If this is genuinely something more general, we should be able to find it in the vertebrate brain, a completely different brain. And remember, we didn't know that the vertebrate brain also had a ring attractor. It was discovered after our paper. And not only do we use the same model, we use the same model with the same parameters. We do not change a damn thing. And we just directly apply it to fish in a schooling context, in the virtual reality. And this is one of the advantages of the virtual reality, is it would be very hard to persuade 2 fish, real fish, to swim in exactly the same geometrical configuration and to consistently look at different geometrical configurations. But we can, in the VR, we can have these 2 virtual fish, which I'm just coloring red so you can see which ones are virtual. And then we predict that just changing this lateral distance, we will see a bifurcation in the brain, in the decision of the animal, where it will average between the options and then choose. And that's indeed what we see. We also looked at 3 options, which took a lot more data, as I mentioned before. And now we still see this double bifurcation. It looks a bit different because now we have a moving frame of reference. Our targets, the virtual fish, are on the move. And so the bifurcation diagram looks a bit different, but you can still see this double branching structure. And again, without the theory, firstly, we wouldn't know what experiments to conduct. Without the virtual reality, we wouldn't be able to conduct the experiments we wanted to. So it really needed both the theory and the technology, because how on earth would you interpret data when you get it? And so again, we found evidence that indeed the brain is exhibiting a double bifurcation. And what we're doing now, as I said, is we're now able to image the brain during these experiments, because that will be the real proof that what we would expect to see on the ring attractor are exotic dynamics at the bifurcation point. And in virtual reality, we can keep the animal indefinitely in the bifurcation point. Even if it differs from animal to animal, we can find where it is, and then as the animal moves, we move the world in such a way that it's still at the bifurcation point. So instead of passing through this point very quickly, meaning the neural dynamics will be hard to explore, we can keep it there indefinitely. And what we would predict is scale-free neural dynamics. And also, we should predict that what we're doing with the flies is we should predict that at the bifurcation point, and only at the bifurcation point, by changing the quality of the options slightly, the fly should basically be controllable. We should flick it back and forth. Beyond the bifurcation point and before, the brain should be much harder to manipulate. So we have these key predictions. We've also looked at things like asymmetric geometries. Most models would predict you're more likely to go to the 2 than the 1. We predict it's roughly equal probability distribution. And the gray dotted line is where we predict the animals on average to actually be. And so we can test these other theories that distinguish our model from others. And the probability distribution was similar and the positions were near identical. I do have this. I didn't remember I put this in. So Armin joined us from Harvard about a year and a half ago. And so our labs are directly opposite, so we're working really closely together. So you can paralyze the fish with Bunga toxin, so snake venom. Then we decode the neural commands to turn left, right. And then the animal thinks it's in a virtual world and thinks it's swimming through this virtual world. And you can have whole brain imaging at neural level resolution. So we're now really working to reverse engineer this whole neural circuit. We've also taken this, and now in Constance, we've applied it to drones, to how drones fly, also how robot vehicles coordinate their behavior, how they respond to one another. And we've been, I don't have the really important figure. Doesn't matter, I'll just tell you about it. We've also been working with Diana Roos at MIT. And she's famous for lots of things, but they came up with the human optimal controller for robots, which is incredibly complicated, takes a day and a half on a supercomputer to optimize for our systems. And we compared its performance to the fish algorithm using the same parameters that the flies, the fish, and the locusts use. And it's within 5% of provably optimal for all of the different systems, including we now have watercraft as well. So nature has found this very simple algorithm that's really effective. And what was also cool is, remember I was telling you before that the left could cancel the right and they all go down the middle. That's what they do in the classic flocking model. And so it made me realize that the classic flocking model is wrong. By studying the brain, it gave me a new insight into flocking. And you need this forgetting rule that I mentioned earlier. When you end up going the way you don't want to go, you begin to forget your original preference. If you add that in, which was in the original Nature paper, but neither I nor anyone else has ever used it again, but we need that to put it back in, and then suddenly the animal group, the fish schools, the schooling model behaves in exactly the same way as the brain. Now what we've done in our paper is we can prove that the ring attractor model can be translated to the spin model. What we're now trying to prove is that the spin model can be fully translated to the flocking model. And then it shows that there really is a mathematical parallel. So we're working on that at the moment. If this is such a robust general finding, then we should also find it in the wild. So we worked on wild baboons in Kenya. We're using GPS. So this is with my PhD student, Ari Strandberg-Peshkin. She's now at Max Planck as well, and Damien and Meg. So here we have, it's quite hard to do these studies, to catch these animals, to put the collars on. And for obvious ethical reasons, we did not put collars on juveniles. All of the collars are removed automatically. And the juveniles tend to follow their mother anyway, so we didn't think it was so important. And so now every second in their lives, we have the locations of the animals. These animals have a strong dominance hierarchy. All males are dominant to all females, and there's an alpha male. And if you look at the natural history shows, it's always like the alpha male tells them what to do. The alpha male dictates this, the alpha male does that. The colors here represent males, females, and subadults, and just looks at a decision where some give up and tend to follow others. Well, we had tens of thousands of such decisions.
When you say second, is that a figure of speech or is that the actual resolution?
Second is the resolution. Yeah, the spatial resolution is within 60 centimeters. The temporal resolution is every second. And so, it's, at least for me, it was kind of surprising that we can go from these theories that were really originally sort of thought of, at least in my mind, as being just about animals moving as groups, to now being able to think about how the brain of a baboon makes decisions when it perceives different numbers of individuals going in each way. But again, we find evidence for the same bifurcation. So it does seem to be a general principle. So the general conclusion here is we find there exists a common algorithm that governs decision making. from the neural dynamics to individual decisions, and from individual decisions to collective behavior. So I think it's really, I don't know, I think it's almost too beautiful to be true, but it is true that there's this deep similarity between these systems. Now, I also realized during the break that I've published 2 scientific publications on soccer. And so I thought now would be a good time for us to switch to collective behavior of soccer. But does anyone have any questions before we talk about football? We also have tomorrow, and we also have the field trip, which I'm about to propose. And so, in this paper, let's see if I still have it. It doesn't matter. I did have a video. So in this paper, we actually developed algorithms that allow us to try to predict where players will pass. So you can sort of work out the sort of areas where the ball is more likely to be picked up by other players. I do have a video. We can show it tomorrow. But I think it's more important that we see a real football game. There's another paper that we published a couple of years ago where lots of predictions about soccer, about football, have been based around the individual. You always hear about Messi and other footballers, if you know something about football. But it's always about the individual. But of course, I mean, Germany was a famous team that won the World Cup with a team that people probably thought didn't have superstars in it, but they played very well together as a team. And so we had amazing tracking data from the Bundesliga, where we have all of the players, we have all of the football, we have it sub-second resolution. It's remarkable data. We're not allowed to tell you which teams they are. We had to anonymize all the players and all this kind of stuff. You can figure out a few of them from the data, I think. And we could do all sorts of interesting things that look at the collective effects, how well teams play together, how well different combinations of players played together. And we could even then use that to see if we could predict the market value of players, both at present and in the future. And could we do so better than, you know, statistics like how much did he run, or how much did, it was all male football, so that's why I'm saying he, you know, how often did they pass? And so we think that these collective, with the advent of the ability of getting these technologies, if you have a deal with the football clubs, you can get these technologies, and there's a lot of really interesting things, but I think it's very important to actually experience this. So I propose that we do a field trip now, and we go and watch a football game and consider collective consider collective behavior. And we can also discuss it on the way, because remember, fish schools and bird flocks, they're basically like fluid computational entities. Being on the move helps them think.